This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 864, recorded on February 8th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello, Vincent. Let me see. I'm sorry. I'm not paying attention here, but it'll be up here in just a second. It's 65 degrees and sunny. We're emerging from a cold snap. Everything's glorious.
It actually went above freezing here in New York for the first time in a few weeks. It actually feels like spring, that I think the Canadian north wind stopped blowing today, whatever, something like that.
It's all McFadden's fault.
Indeed. Also joining us today from Madison, New Jersey, Brianne Barker.
Hi, it's great to see everyone. It is in fact 37 here, so balmy for this time of year, but it is very cloudy.
Today we have a special treat for you. We have a number of guests who've been on before and a new one added to the mix. And let me start with from Queensborough Community College, Monica Trujillo. Welcome back.
Thank you very much.
Nice to see you again. It's been a year since we had all of you on, so we're going to hear what you've been doing in the past year. From Queens College, John Dennehy. Welcome back.
Thank you. 44 degrees Celsius here and also very cloudy.
It's pretty warm, right? It really got warm today. Last time she was here in New York City. Now she's joined Rich Condit in Texas. Davida Smith, welcome back.
It's great to be here again. I'm happy to be joining y'all today.
So last time you were at the new college, right? Is that the name of it?
The new school.
The new school.
New School University. Now Texas A&M and San Antonio.
So you could have been a few blocks from the incubator.
I know.
But instead you're thousands of miles away. Oh well, such is the way.
What was I thinking?
Rich, how far is San Antonio from you? A long ways.
About an hour drive.
Oh, yeah, not bad.
We need to hang out.
Yep.
Right.
We can do that.
And also joining us for the first time from the University of Missouri, Marc Johnson. Welcome back.
Thanks for having me. This is a long-time listener, first-time visitor, but 55 and sunny. We're almost melting the snowstorm from last week that shut down the university for 3 days, but it's really nice.
What did you get, a half an inch?
Yeah, about that. No, it was 6 or 8 inches. It was a good Ithaca flurries, which is where I lived before here.
Ah, okay. I'm sorry. I said welcome back, but this is your first time on TWiV. But we learned that you're a long-time listener.
I've been on before. I've been listening so long.
Since the beginning. Very good. All right. Well, welcome all. The last time the 3 out of 4 of you were on was TWiV 737 in March 2021. So it's about a year ago. And we've had a lot of new listeners in that time. What was the? I think it was SARS-CoV-2 in New York City wastewater. That was the name of that episode. And I actually found a picture of the waste processing plants outside of the city that I used for that. I found it somewhere online. Those domed things, right? What are they called?
Yeah. Egg aren't they the eggs?
The eggs,
Yes.
right?
Yeah.
How they are called.
So that's one of the waste treatment plants, right?
Newtown Creek.
And what's the number? Does it have a number?
Are we allowed to say, Monica?
No, you're not allowed to say a number. Because in your paper, you say WWTP, wastewater treatment plant.
We're not falling into that one.
But it's in the paper. It's published, no?
No, we can't say what plant it is.
Oh, that particular one. But it's one of those WWTPs, right? How many of them, how many total are there?
14.
14. Okay. Let's just review how this got started originally without Marc, and then we'll move into your new paper, which just came out, tracking cryptic SARS-CoV-2 lineages detected in New York City wastewater. Monica, do you want to tell us how this got started?
Okay, so this started with me reading a paper about the detection of SARS-CoV-2 in an airport, and a phone call to my good friend John Dennehy, who has a lab that has the biosafety level where this research could be done. And we reaching out to anybody and everybody that we knew, trying to get the New York City DEP interested in this, and having the good luck to being able to contact Pam Elardo, who is somebody pretty high up there, who immediately was interested in giving what she called CUNY an opportunity. We found out that there were other people that were interested in analyzing wastewater in New York City. And we were, I think it's reasonable to say that we were the backup plan, but the backup became the main one, as our protocol was the one that was used. And we had, from the very beginning, had the idea of sequencing this wastewater genetics, this genetic signal from wastewater. But we didn't, we had Davida who had, started, and we were wondering how we were going to continue this. And that's when, the conversation with Marc happened. So we had already collected some data. And then we started working with John and continue some of the sequencing being done by Davida. Her moves made things a little bit more hectic for a while, but now, things are back.
So you were able to get samples from each of the 14 plants, right? Originally,
Yeah,
way
once
back.
a week.
Once a week.
Once a week.
And if
starting in May of June of 2020.
Okay, and then if I remember, you had to develop some protocol because it's not straightforward just to extract from waste, right? This waste. And so who did that? Is that John and Davida mixture? John, go ahead.
Well, previously we had done work where we isolated bacteriophages from wastewater. So
right.
It's a natural switch for me to say, why not use the same protocol that we use to isolate bacteriophages to isolate SARS coronavirus? We tried it. It worked great.
Could you briefly review, I know we've done this on the last episode, but just in a few words, could you briefly review what that protocol is?
So the protocol is essentially we use polyethylene glycol and salt to precipitate the virus. We centrifuge it, we filter it, and we kind of go old school, and then we use trizol to extract the RNA. Other people tend to use the, QIAGEN kits. We like the old school way. We had some good success with it, so we continued using that. And once we extract the RNA, we use the CDC kit to assess the frequency of the N1 gene in the wastewater using quantitative PCR.
So, Davida, did you do the PCR in your laboratory? Is that right?
So this is the thing. So it was properly, we all worked together in this one. So when we were thinking about the sequencing strategy, we were looking at what everybody else was doing, and everybody else was doing kind of whole genome approaches using kind of Illumina and Nextera type sequencing libraries. And we thought that a targeted approach made more sense. So in essence, we used what we knew, same exact thing. We knew that we could use sequencing like we were doing for the microbiome that we had been doing all along. So I've been doing microbiome work with my students. So I said, why don't we just take the same strategy, stick the adapters onto the end of the primers for spike, and then do that. So Monica and Sharon, the postdoc over in John's lab, they designed the primers, and then we made a strategy whereby at the start it was I was doing everything, but then Monica would amplify. This is how crazy it was. Monica would amplify the spike and then clean it up with beads and then bring me the amplicons, and then I'd sequence it. So we were able to do it kind of in tandem. So poor Monica spent her life driving around the city, getting the samples from Queens or from the wastewater plants, bringing them to Queens, doing all the isolation and all that work, and then bringing the amplicons to me and me sequencing them over it at the new school. And the thing about the new school is like, we didn't really have a very big science department at the new school. It was really just me. So I was there on my own. So the only human contact I had other than my partner was when I'd get samples from Monica. She'd drive up with a cooler and then hand over samples to me, and the occasional box of tips and things that we needed. So, but it was a proper, like, all of us putting our minds together and trying to solve a problem. And, John says the protocol was really easy, but there was a ton of optimization and figuring things out that had to happen before the protocol was really working. And that was all the work of the grad students and Monica and everybody over in the lab doing a lot of replicates.
You remember when we added crystal violet so people
Absolutely
could see where the precipitate was, and we film it and we send it to the DEP lab? I mean, there are so many stories.
And the thing is, like, things that we would take for granted. So, when they were tipping out the centrifuge samples, they were losing pellets, and that's where that came in. So then we had to come up with ways that they could resuspend the pellets with glass pipettes and things like this. So there was tons of little itty bits that we had to kind of optimize to get it to work. And then the joyful thing was that the DEP actually started to use the protocol that we were suggesting. And Monica is lead author on the protocol paper that's in PLOS One.
So just for sort of clarifying, I know we talked about this last time as well, but the we that you're talking about includes some students. Is that correct?
That's exactly right.
And can you talk a little bit about kind of what level students and what types of things they were doing?
I'll let Monica take that one.
So essentially, we were not allowed to have undergraduates in the lab. So I had to go find all my former students who had graduated. And, fortunately, some of them were taking gap years because no one wanted to go to medical school during the pandemic. So they all came back to the lab, and since they had a BA, they were allowed on campus to work despite the shutdown. And we trained them. Well, they didn't need much training. Fortunately, they had quite a good experience with working with bacteriophages. So training them in handling coronavirus was not that big of a stretch. I owe my students Zach Peng, Matt San, Kristen Cheung, Anna Gao, Nana Kubata, Michelle Markman, Shannon Rai. All of them have been working on this project since probably June, and they've done a tremendous job, and they don't nearly get thanked as much as they should for this. PIs like me and Monica get all the recognition while they're doing all the work behind the scenes.
Yeah, I agree 100% with John. 100%.
I assume that there are in this protocol there are biosafety issues for a significant fraction of it. At what point does it become safe? Is that the RNA extraction or what? How do you deal with this?
Well, we were not allowed to open the bottle until we had pasteurized it in the lab. So we would get the sample from the DEP in 250 milliliter bottle. We bring it back to the lab, put it in a hot water bath, and heat it 60 degrees Celsius for 60 minutes, inactivate the virus, and then only then would we open the bottle or start working with it.
So the pasteurization process, does that leave the particles intact? Are you still precipitating particles with PEG, or is it just nucleic acids?
It's a good question. We don't quite know the nature of what we're detecting, and that's something we would like to follow up. But we had looked at the effectiveness of comparing unpasteurized with pasteurized extraction. And actually, the pasteurization process seemed to improve the recovery of RNA. And this is something that's also backed up with a couple of other labs that have done the same thing. We reported this in another paper. It was like comparison of 36 different labs that had processed wastewater from Los Angeles. And, they used a wide range of different techniques. And then they looked at, who was the most effective at, extracting RNA from the same sample using the N1 primase to detect it. And, the protocol that we used was one of the most effective at isolating and concentrating the RNA.
Okay.
Just to throw this out there in case there's any wastewater people out there, we found the same thing, but you should be aware that after you pasteurize, the half-life gets much shorter. If you're going to pasteurize, use it right away. Okay. And also don't freeze it. If it's wastewater, that can kill the signal.
So
very good point.
these
So-
Things we learned the hard way.
have always.
Yeah, so it's interesting. If you recover it and you store it at 4 degrees, it lasts quite some time. Probably could be up to a couple of weeks. Whereas if you freeze it or you pasteurize and then let it go, then it just decays.
And we should mention from our work, it is envelope particles we're detecting. If you concentrate them and run them on a density gradient, they have the exact same density as HIV and envelope particles. So, and if you treat it with detergent, the signal goes away. So we're pretty sure it's envelope particles.
So the 14 wastewater treatment plants, they each serve a different part of New York City, the city, right? All the different boroughs, they're throughout Manhattan, Bronx, Queens, Brooklyn, Staten Island even, is that right? Yes.
Yep, Staten Island too.
And they collect, obviously, sewage from homes, correct?
That's correct.
Do they take sewage from the catch basins on the street as well, or is that going to the rivers?
So it's a CSO that we have in New York City. So pretty much all the water.
What's
that's above ground.
CSO?
A combined sewer overflow.
You can't say we anymore, Davida, sorry.
Say that again.
You can't say we anymore, you're gone.
Oh no! Don't say it like that. Don't say it like this. That's brutal.
There's one
that doesn't. Yeah.
Yeah, that's the one in the south of Staten Island.
All right, so it rains, goes into those grates. There's a pipe system, and so some of that, I mean, I used to live on the east side of Manhattan, and I know that there are pipes that, there used to be anyway, 20 years ago, that go from the catch basins into the river, but some of them go into the sewage treatment plants, right?
Yeah, a lot of them still go into the river.
Yeah.
They go in, like
I gave Monica a fight when we went to the combined sewage outflow at Flushing Marina, which is, sort of quite close to Queens College, and we wanted to see if we could detect the signal in the outflow. And I climbed down basically into the pipe, and I disappear from view, and she doesn't know what happened to me, and she imagined the worst. And then later on,
that was terrible.
Bottle of wastewater.
I just imagine John's wife telling me how you could let him do that.
That's right.
John's had some pretty heroic efforts in this project.
So that's where we left off last time, and if I remember, you had seen the RNA kind of paralleled what was going on in people, right, in terms of levels of the outbreak. Is that correct?
Yeah, and we had identified the mutations that were not seen in people at that point. So we already knew that there were mutations that we couldn't necessarily explain, but we didn't have a tool yet to kind of pull out linked mutations, and that's where Marc comes in, because he was the first that was able to show the linkages of the mutations together.
So at the time when we first had you on, it was early in the variant parade, right?
Yeah.
Was Alpha around? Did you pick up Alpha? I don't remember.
Yeah, we had Alpha.
Was around at that point, right? No, you're right.
It was
2020 with Alpha.
21, yeah.
2021. Okay.
So it's interesting. So we had used a targeted approach, and we had come on the show to describe, how we had started sequencing. In the back of our minds, we had known about these kind of weird mutations, but we couldn't really put them together and quite understand them. So I don't think we actually talked about them on the show. But Marc had seen the show, had realized that the two of us, the two groups, had kind of converged on the same solution, which is to target the receptor binding domain specifically, because that's where the interesting stuff is happening. So immediately after watching the show, he reached out to us and said, we should get together and compare notes. And then
there was a little more to it than that, but yeah.
Okay.
Were you doing similar things, Marc, at your place?
I was doing almost exactly the same thing.
Okay.
It's just everything he had was bigger.
I still remember that. So I watched your TWiV dropped on April 1st, and I sent John an email the next day. And I remember that the subject line was convergent evolution.
Yeah.
Everything we were doing was just so similar, and it was unique because almost no one else in the country was doing it this way.
So, Marc, where were you sampling?
So shall I give my
go ahead
now? Okay, so I'm at University of Missouri, and I am a purebred virologist by training, molecular virologist. I had never considered environmental sampling in my life until April of 2020, where I got an email out of the blue that the state, the Department of Natural Resources, had reached out to the health department and had heard about wastewater testing and thought it'd be a good thing to set up. In Missouri, we already have a system for wastewater samples being transferred to a central lab. So we already had this sort of infrastructure in place, but they didn't have the infrastructure to do the sampling. So they were reaching out to different groups and saying, hey, can you do some PCR for us? And they said, do you know anyone at Missouri who can do it? And I'm like, I'm sure I can find someone to do this, but I couldn't find anyone who could do it. But I didn't feel like we should be saying no. This was a pandemic. So I just said, go ahead, we'll figure it out. And I found a colleague in the School of Natural Resources who actually does know about environmental sampling. And we started working together. And then starting in May, we started getting samples from Missouri, which is at first it was 7 samples a week, and now we're up to, I think, 200 a week that we get. But
wow.
So then through 2020, it just kept growing, and everything was on a public dashboard, and it was pretty well received. But through 2020, it kept coming up in discussions of whether people were going to start sequencing the genomes. And I think I agreed with probably a lot of you on this podcast, even if you don't admit it now, but I didn't think there was a point, because I thought the virus was changing so slowly that there would be no point to this. I mean, I remember Vincent, that you made the point at one point, the virus only has 7 mutations throughout the world so far.
Yeah, that's right.
I thought exactly the same thing. It's like, this guy just is not accumulating mutations. It has the lowest error rate of any virus ever. What would be the point of sequencing it? But I can tell you the point in time when I changed my mind, and it was from TWiV. It's when in December you did the preprint from Jesse Bloom's lab on 229E, where he was looking at the evolution of that over time and very clearly showed that it was adapting, and it was all in the receptor binding domain. And this had consequence. It was making it resistant to serum over time. And it was just like a light went on. It's like, okay, I was thinking about this completely wrong. And so I went to the lab and I'm like, okay, how can we do this most efficiently? And so I read his paper really carefully. And fortunately, the receptor binding domain is less than 100 amino acids. And we'd already done a lot of work on a sequencer called a MySeq, which has the advantage that you can do paired end reads, and it can go 300 nucleotides from either direction. So collectively, you can get about 600 nucleotides of one continuous read, which is enough to cover the entire receptor binding domain. So what we did is just designed a primer set that amplified a 550 base pair piece, and then we would sequence it on the MySeq, and then it took some doing to figure out how to sort out what is a PCR chimera and what was real, but it once you get the software in place, it's very straightforward.
Where were your samples coming from? All over the state, or?
Yeah, it was actually very interesting. We started just before the Alpha variant arrived, and so we kind of could trace how it was passing through the state. We saw first the Alpha variant arrive, and then July, we saw this really interesting development of the Delta, and then most recently the Omicron. In each case, we could sort of track how it spread through the state. It's pretty cool.
Marc,
you see it in like Branson, Missouri first, and then you were able to trace it along some expressway all the way up to St. Louis.
For whatever reason, the Delta wave started in Missouri, and the first place we detected on May 6th was from Branson, Missouri, this little tourist town. And unlike the Alpha and Omicron variants, which sort of followed along the interstates, it sort of would follow the population centers and then work its way out to the rural parts. When Delta came, we saw it in Branson, and then we just started seeing it randomly appear throughout the state in like rural parts of the state, which we assumed were people that had taken weekend trips to Branson and brought a little present home. It was interesting to watch, though, and it happened so fast.
Marc, before you got into this, what virus were you working on?
Nominally, I was a retrovirologist, so I did my PhD my postdoc with Volker Vogt at Cornell, and I use retroviruses as kind of a tool. Really, what I've studied for most of my career has been pseudotyping, trying to understand the rules of why some glycoproteins work with other cores and others don't, and trying to figure out those rules. So I work with HIV, but I have a panel of scores of glycoproteins in my freezer that I played with to try and figure out, okay, why does this one work and this one doesn't, and stuff like that.
I see. Now the paper is making sense. Yeah. We'll get to that. Okay. So you shot an email to this group, and what happened?
So I need to back up a little bit.
Sure.
So we started sampling, we watched the alpha variant, it all made sense until the beginning of March, when there was a single sewer shed in St. Louis area. It was one of their suburbs, that it had what first looked like a California variant. It had the L452R, but then the next week it had a second mutation, which was N460K, and it means nothing. But then something freaked me out the next week, is that we saw the same combination, but we actually saw N460K twice, because you can get the same amino acid change through two different nucleotide changes. So 2 parallel nucleotide changes leading to the exact same amino acid change, all in the same lineage, and only in this one sewer shed in Missouri. And this mutation had hardly ever been seen in patients. And it gradually was picking up more and more mutations. And, looking back now, they were not random mutations. N440K, which is in Omicron, 417T, which it's K417N, which is it had a lot of mutations, the same place that now are Omicron. I didn't know what it was. It looked to me like it was a patient who had been who was maybe immunosuppressed and had gotten Eli Lilly treatment with the monoclonal antibodies, because based on the antibodies, when we confirmed this, they were completely resistant to those monoclonal antibodies. So that's what I thought it was at first, but I wasn't ever sure. But I was crawling out of my skin trying to figure this out. It was nowhere else in the world. It was not in any patient, but we were seeing it in spades from this one sewer shed. So I started reaching out to everyone I could think of who was doing wastewater work across the country, and just like, is this just me, or are other people seeing this? And they all said the same thing. I don't know, because everyone else was doing whole genome sequencing, where you break the virus into little pieces and you know what polymorphisms are there, but you don't know if they're linked or not. So they can't really tell whether there's something new or not. But the way that we were doing it with this targeted approach, we only get 5% of the genome, but we get it with great depth, and we know which mutations are linked. So, going back to when I heard your podcast about Jesse's paper, I'm like, okay, I don't know if this alpha variant is really a thing, but I now know that there will be mutations, and I don't know what they will be, but I know this is where they will be. So, I was kind of thinking from the beginning, let's design a strategy that will find what's coming and not just what we know about. But then, and then I heard your podcast on April 1st, and it was so exciting because they were doing everything exactly. They had exactly the same thought process I did. They were using the same purification techniques. They had the same targeting approach. And so, like I said, I heard the podcast on April 1st. I wrote to John on April 2nd. I think we had a Zoom call on the 3rd or the 4th. And within a week of when the podcast dropped, I had the first samples being sent to me from New York, because as we figured out, we had the same approach, but we had a way of doing a slightly larger chunk. And we also had gotten a little further along with developing the pipeline for making sense of the data. And then initially it was going to be a one-off that I just wanted to see if they were seeing the same thing I was. And if there wasn't, we probably would have dropped it there. But already on the very first pass in April, they had a few of these lineages. They were at small percents. They were only 4 or 5% at the time. And they were not the same as in Missouri, but they had the same clear, like, this is not random. They had all of these mutations in places that were known to be part of the receptor binding domain or were antibody escape mutations. And we were like, okay, we got to keep going with this. And so that was beginning of April. Within 4 months, we had submitted the publication that finally came out a week or two ago, which feels like a lifetime ago.
Hate-love relationship with FedEx.
Yes.
and the weather.
Yeah, and the storms.
Yeah, exactly.
Okay, so let's talk about what you found in New York City. So it's all in the paper, pretty much, but who wants to start that forward?
Well, essentially, since the beginning of the year of 2021, we've been witnessing what Marc had called oddball variants, and they just have really unique combinations of mutations, unique constellations of mutations that never show up anywhere. I remember probably February of 2021, we got some of our sequencing data back, and there were a couple of mutations that showed up a few times. Q498 and Q493.
Wait a minute, John, you didn't know it was Q498Y yet. Remember, because you saw Q498H and Q498 stop code, and you thought it was an error because there was a stop code on, but it was because they were both in the same sequence.
That's right.
That was the big key. That position was the real key.
Right.
But at the same time, you're picking up the prevailing VOCs at alpha.
Yeah.
I think you go alpha, beta, epsilon, iota, delta, kappa, gamma.
Yeah.
You saw them all, right?
Yes. Yes.
And we were able to see changes in the frequency over time as well.
All right.
Yeah. But what's interesting about the Q498.3 and Q498Y is I just Googled the mutation, and the only things I could come up with were a couple of papers. I think one was from Ralph Barrick's lab where he adapted the virus to mice.
Right.
And then there were a couple of Chinese groups I think did the same thing.
Right.
And every time you see the same mutations appearing.
Yeah.
And at that point in time, Q498Y had never been seen in a single patient. And I think to this day, it's never been seen in a single patient. It's extremely rare, at least in humans.
All right. Now you ended up looking at four lineages, right? In New York City.
To be clear, those are the four that were most consistent and best understood. We see more lineages, we think, and they kind of pop in and pop out of existence over time. Some of the ones we saw early in 2021, we no longer see. Some we still see. I think there was one from one area that popped up, I think, in April of 2020, and then disappeared until just very recently.
Yeah.
So you mentioned that many of them
it added on a whole bunch of new mutations on the same genetic background.
You mentioned that some of those popped in and popped out. So how long did the four that you really focused on persist?
They've been there. They've been with us the whole time.
Some of them we haven't seen in a little while.
Yeah.
It's harder to find them when there is a wave. Sort of early summer was the golden period for us because it was between the alpha and delta waves. So the patient levels are really low. And we think that's why during that time, some weeks, these were the only things we were detecting from the wastewater. They had become the dominant lineages.
And you say as the
Omicron wave, it just kind of totally dominated everything we saw. So we actually designed a strategy to eliminate Omicron from our samples and just detect anything that's not Omicron. And that's when we were able to see them again. Plus, I think we were able to actually, by accident, detect B2A.
So you make an interesting comment in the manuscript. You say as the human signal decreased, right? There were troughs in the outbreak. The signal of these increased, or right?
The percent of the relative percent increased.
Yeah. So presumably the amount is the same, but it's becoming a larger fraction of the total, right?
Exactly.
And I guess that's a hint that it's coming from something other than people, right?
Or if it's a human population with a constant input,
such as
an immunocompromised patient with a really raging GI infection that lasted for a very long time.
Do you think you would need...
I know, nothing makes sense. I'm
just, how many would you need? Is it one? Would you think there would be one in Staten Island, or would you need multiple?
In my mind, it only makes sense if it's one person. Because it's, it would have, because it's changing over time sort of together. It's hard to imagine that as a community.
Yeah.
If it's a community, that means it's contagious. If it's contagious, why are we only seeing it in these, geographically constrained? But if it's one patient who sheds a lot of virus, then it makes sense.
Super shedder.
And there have been cases of people that have been, I think you reviewed one of these, or you mentioned it in one of your, in the one where you're talking about origin of Omicron, of someone who had it for a good 9 months.
Yeah, that was an AIDS patient in South Africa who.
She only shed it because they gave her the therapies to get rid of it.
Yeah,
I mean, there's been patients over a year. So it can happen. It's hard to believe that they're shedding so much that we're detecting it in a sewer shed with over 20 million gallons a day of sewage, but it's not impossible.
Okay, that was going to be my next question. So you sort of answered it.
I mean, so I don't know about you, but I have never seen good evidence that this virus is reproducing in the gut tract. Okay, I think my feeling is it's passing through swallowed mucus and so forth. To correct me if I'm wrong, but
No, there have been people that have shown it, and they've taken gut biopsies and shown.
Yeah,
it.
I mean, a few particles here and there, though, that's not really robust evidence for gut reproduction. Okay, it could, because in bats for sure, the ancestral virus probably is a gut virus, so it's not impossible. But EMs of a couple of particles, it's not telling me.
No, not EMs. They've done an immunofluorescence of
yeah, even that,
but the opposite makes no sense either, because these are envelope viruses. They would have a really hard time surviving the stomach and bile.
Not coronaviruses. There are animal coronas that go through transmissible gastroenteritis virus, right? TGEV. It's a corona. It goes into the gut. And that's always amazed me, because I used to teach students envelope viruses never go through the gut intact, but here they are. So you throw out your binary stuff. There's no black and white in biology.
Right, but if it's true, that is a maddening fact that you are putting your thumb on, that there are these examples.
Yeah.
But they don't make any sense, because we know that bile can kill these guys, and there's a lot of bile.
Yeah.
And incidentally, the places where they see the infections are usually downstream of where the bile is in the GI.
Yeah. I mean, you don't have. I don't think you need it to be reproducing in the gut to get your signal, right?
No.
It's just passing through. You swallow a lot of mucus in the course of a day, right? That's
possibly. Although they've shown that the shedding, the fecal shedding, lasts longer than the
nasal secretions, nasal shedding.
Yeah.
But we also have 4 distinct variants as well. So that's important to understand, that it's like, if it is a person, it's 4 individuals.
More than that.
4. But you know what I mean? That's what we're saying.
4.
It's going to be at least 4 different mechanisms, if you like, that's responsible for all of this. So,
are these unique to each wastewater treatment plant? I think I remember that, right? Each plant has its own unique variant. They don't seem to transmit.
No.
So that's why we're saying the mechanism might be completely different in all 4. We have no idea.
But, we see 2 different variants in some places, 2 or more, right? That are consistent, that are very different from each other, and come from the same source. So, it's really hard to understand what's going on when you have so much unknown.
And I think that something that we have done is we try to get in touch with GI people to see what was their understanding about not only the biology of the virus in the gut, but had they sequenced viruses from feces and have found any difference. And really, there's very little data. We had some interesting conversations, but no way to get to anything that, at least to us, could help us to understand. They were interested, but most people didn't have the funding. Most people that were studying the virus were focusing on the respiratory aspect of it. So really, we did try. I think we spoke with like 4 or 5.
Exactly.
But we couldn't really get really anything that could help us to understand it.
And ideally, you want to have fecal sample and detect the virus in those fecal samples. We weren't able to get access to any fecal sample.
No. And even having access to the labs to even do the work. So folks are very busily working on human stuff as well. So any work on like infectious wastewaterness was not something we could even do.
So just to be clear, these 4 lineages, what you call cryptic lineages, right? And cryptic means they're not in people, right? They're not in the database in people that you can tell.
Not in the database.
So they are unique to New York, right? Don't find them anywhere else.
Not that we found yet. Not yet.
Well, no, that's not. Well, I mean,
California.
We saw them first in Missouri. There was actually 2 sewer sheds in Missouri, because there was another one that very briefly had one in the summer. And then very recently, there was a group in California that had adopted our protocol that also found it in one of their sewer sheds. Not the same lineage, but, it's same sort of mutations at the same site, just different combinations of them.
All right. But never the exact same combination of amino acid changes, right? Okay.
But this also gets to the point that basically this has been examined in New York City, Missouri, and California. So you're not saying that this is some sort of special New York City mechanism. It's just that you happen to be the ones who have found this. And if people looked in more places, who knows?
That's what
I've
we're
looked
thinking.
in as many places as I can get my hands on samples, and it is rare. And we should mention that the NIH contracted this company BioBot in July, August, that was doing nationwide sampling. And part of it was to do sequencing, which they immediately dropped in NCBI. And my lab, we've gone through those sequences pretty carefully to try and look for combinations of mutations. And mostly what we've done is rediscovered the New York mutations about a dozen times. But the problem is also that, everyone can be unique. It could be that there's more out there that we just didn't have the right combinations. We're talking about screening through terabytes of data here, so it can be a little tricky. There were a few others that, like so very early on, there was a company called Aquavitas that had some sequence that they deposited that also appeared to have lineages, one from New Jersey and one from California, but it was one-offs, never followed up on. I think there are more of these, but New York does seem to be a hotspot.
Okay.
So I understand correctly that this number 4 variant, WNY4, has a boatload of changes. Is that right?
Yeah.
We've had a few like that. And when I first generated, I don't know if we'll get to this, but when I first generated that, I was wondering whether it would even work. It had a deletion in the receptor binding domain. Who does that?
Right.
But it worked great.
It's a new genetic term, boatload.
Yes.
Well, I use another B load most of the time.
So tell us now. Then you decided to ask if these RBDs were functional, right? So tell us about that.
Well, so like John said, that when we saw the Q498Y, I think John and I both did the same Google because I didn't find it in GISAID. Is it GISAID or GISAID?
Whatever you want.
I thought it was GSAID.
It wasn't in their database, but let's try the bigger database. Let's try Google, and that's where we discovered that these are in a bunch of rodent adapted ones. I'm like, well, I wonder if these can now infect rodents, because it wasn't exactly the same as what Ralph Barrick had used. I actually wrote to Ralph. I'm like, will this work? And they did a little modeling. They're like, maybe. They couldn't say for sure. So then I'm like, I need an ACE2. And then Vincent, it was from your old buddy, Jeremy Lubin, who had the ACE2 receptors and sent them to me. I'm like, can you please FedEx them today? But he sent them to me, and we made the stable cell lines, and so I'm good at making pseudotypes. So we made lentiviral particles, and we introduced the receptor binding domain from these oddball variants. And then we tested, which ACE2s can they use? And with the original 2019 lineage, it can only infect human. It cannot use mouse or rat. But every one of the New York variant lineages, and I should mention all of the New York lineages have had a mutation, one or the other, at position 498. And every one of them was able to use mouse and rat ACE2 just fine. Although I have to point out, that's not that impressive. A lot of the new variants of concern can also utilize those receptors, but it's at least consistent with the fact that the notion that these could be coming from rodents.
Got it. All right. So what kind of animals live in New York City that might be candidates, culprits maybe?
So this is interesting. So we wondered exactly, if we're seeing an animal reservoir, what animals can we detect in our wastewater?
Mm-hmm.
The answer was quite surprising to us. So we sequenced I believe 12S RNA from wastewater. And then we blasted them again, GenBank to see what species would pop up. We got a whole bunch of interesting ones. First of all, humans completely dominate, obviously. But then we saw quite a bit of cow, pig, sheep, salmon, chicken, goat. All of these presumably are not living in New York City. They're from the food that we eat. So that, to me, at least, that was a big surprise that you can actually detect what we eat in our wastewater. But then we saw we saw a few species. But before I tell those, I just want to mention that in one sewer shed, we suddenly saw sea lions. And we're like, what the heck? we realized it from Coney Island, it must be from the New York Aquarium.
Yeah, yeah.
We got a lot of tropical fish as well.
Huh. Cool.
Yeah.
It's
And some
amazing how
zoo animals.
sensitive this all is.
Yeah.
Oh, it's so
Yeah.
cool. I started collecting water just everywhere I could find it. Like there's the goldfish pond on the edge of campus. We did that one. I pulled out like 8 birds and all of the fish in there. And we took a sample from the Missouri River once, and we pulled out like 40 different species of fish from this little quarter cup of water. It's so cool.
Yeah, now I want to go have a conversation with the ecologists in my department.
They'll call it eDNA. That's their own thing. It's a whole thing that they do.
They do it in streams to see what fish are in there for fishing and that sort of, right? So aren't there some chickens in New York City still living?
It could be. They say they have wild chickens or even live poultry.
There are some wet market-like stores.
You didn't pick up any geese, right? That's interesting because
we did pick up geese. We think it's probably coming from ponds, right?
Yeah.
Geese and duck.
I don't know if you heard this episode of TWiV where we talked with the high school group that were sequencing
Christine Maritzi.
For birds, right? And so they got
virus hunters.
Seagulls and geese, I think, were the main ones there. Yeah. But okay.
Pigeons?
Pigeons.
Yes.
Yeah.
Didn't see them very much.
Pigeons were very rare. We saw maybe 2 or 3 times, but it was very rare. So the ones that appeared consistently and at a relatively high level, I mean, obviously humans completely dominated, but the next level down would be rats, cats, and dogs.
Mm-hmm.
And those just kept popping up over and over again. So that's what, that probably our top suspects if we're going to point the finger at any animal.
I love these 2 sentences in your paper. One says that both cat and dog populations were dwarfed by the New York City rat population, which is estimated to number between 2 and 8 million animals.
It's remarkably difficult to get a big number on that, though, because we don't have any rat census.
Well, except that later on you say the lowest rat densities in the city based on the volume of rat complaints received.
Yeah.
Yes.
Okay. So that's how you do the rat population, right?
Yeah.
Complaints.
Just where you can see them.
But you notice in some of those sewer sheds, the rat ribosomal RNA made up more than 1% of the total sequence. And you think about if a rat weighs maybe 1% of what a human weighs, you do the math. And that's just the ones that are contributing to the sewer shed.
Well, the first thing that comes to my mind there is that's where the rats live. Is that right?
Some of them.
At least part.
Some of them.
Yeah.
In the pipes.
So
they
actually.
I think glamorous part of our research was a trip that we took during the summer. John, Sharing, and a new undergrad that had joined the lab, working across the city, collecting rat feces.
Oh, great.
And some of the conversations that we struck with people on the street, I wish I had recorded.
So go ahead.
I went to some pretty sketchy alleys in Brooklyn.
Do we want to go down that road of trying to?
We shouldn't talk about where.
No, the rats in New York are part of our day-to-day life. We just see them every day. You see them on the subway. They're running across the platform every day.
But there were some WWTPs where you had a very low rat signal or no rat signal.
That's exactly right.
Yes.
That's the part that throws everything off.
Yeah.
Are they in places where there are no rats? Are there fewer rats in some boroughs?
Yes.
Yes, but that doesn't necessarily correlate with places where we saw.
I see.
Variants. That's why we think it's very possible that it's completely different mechanism at all 4 sites. It may not just be one vector. It may be multiple possibilities.
Yeah, that's important. I've talked about this before. Is that we tend to look for a single solution, and that can be a real distraction.
Mm-hmm.
Yeah. So to this point, there are 6 sewer sheds where we've seen reproducible lineages, each unique from New York.
But not all have high rat populations, right?
No.
No, some definitely have some really high rat populations.
Yeah.
That's not. Some don't. I think at least one of them, Staten Island, probably doesn't have a very high rat population. And just to have a shout out, Richard mentioned this. We look for the rat complaint. That's actually data that's compiled by the city of New York, and they make it available You can look graphically online to see the region where the rat complaints are highest densities. So I would, get my Google Maps out. I would find out where the highest densities were, and then I start walking around the alleys in that neighborhood and look for the rat feces that usually found around the trash cans. And I put my gloves on, make sure I have my mask on because there are some really nasty diseases in rats, SARS coronavirus notwithstanding. And then I would, pick each one up, each little feces pellet, put it into a plastic bag, label the bag with, geographical coordinates where I found it, put that away, and then go down to the next set of trash cans and look. I spent hours doing this, and we never got I don't believe we got a single really strong hit from the feces that we collected. In addition, we worked with the
APHIS.
USDA.
Yeah.
APHIS. I don't recall what the acronym stand for, but the Wildlife Disease Unit, and they would come to New York City and trap rats. I didn't myself feel comfortable doing this, setting some traps up in Central Park or Prospect Park, and coming back later and collecting dead rats or worse, live rats.
They'd bleed them and send me the blood so we could check to see if they had neutralizing antibodies, and nothing clear.
I think it's a low frequency event. I mean, there are a lot of rats, right? Millions of rats. If you're just trapping a dozen here and there, you're not likely to pick up positive ones unless they're all harboring
a rat needle in a haystack.
What about mice? You didn't talk much about mice, but there are probably a lot of mice in the city, right?
No.
Strangely, we never picked up.
See the signal.
You didn't see a signal, huh? Interesting.
Is that because mice are too small?
It could be. They have a very small, the grain of a rat feces is probably maybe the size of a chickpea, where the mouse is probably less than the size of a grain of rice. So, maybe we're just not picking up the signal. The other thing I want to note is that you've heard the reports of, SARS coronavirus in deer, and it seems like any deer they pick either had the antibodies or is currently infected. So that's kind of an interesting contrast to what we're seeing. Is
there any overlap in the amino acid changes between deer and yours?
No. There are hardly any.
No, so that's the really interesting thing. Is anytime they pick up anything from deer, it always seems to be the currently circulating strain. When Delta was dominant, the deer had Delta. Omicron is dominant, deer have Omicron. We don't see the sequence of mutation that we're seeing in the wastewater.
Those just dropped in the last week. If you haven't seen it, there was some deer from Staten Island, and they had Omicron.
Yeah. One thing that struck me when I heard your episodes 2 weeks ago is that when we look at wastewater, we are looking at a huge number of people, huge, right? And we look at it every week, right, across a year. And then we have, like in our case, we tested 100 rats. It's nothing.
Yeah, it's not even possible.
So when we are looking at wastewater, the amount of people that, the amount of virus that we are getting from very, very different sources, it's really, I think we are not comparing, even with the deer, that I'm not saying they have, any mutation, but I'm saying that the numbers of sequences available doesn't compare to the opportunity to see variations when we look at wastewater.
This may be biased because I'm on a college campus in New Jersey, but you also, I don't see anything about squirrels and chipmunks here either.
That's true.
I know we can detect them though, because some of my river samples had both red and gray squirrels in them. But we didn't see them, not to speak of, in the New York City sewer sheds.
So in the end, you don't know if it's an animal or humans or both, right?
No.
Bottom line. So you're going to just keep looking, and maybe at some point you'll pick up something that ends up in people, and then maybe you can say it was there first, right?
Well, it seems worthwhile to figure out where this reservoir is, because wherever these guys are, they seem to be under the same general selective pressure that Omicron was under. And as you just covered, we don't know where Omicron came from. So we don't know where pi might come from. There's lots of surveillance every year to look at where the next influenza strain is going to come from. I
Yeah.
assume at some point the same will come from Omicron, but before we can do that, we have to figure out what the possible sources are.
That's a good point. There's a huge influenza surveillance network globally, and I guess we need to set up a big one or tap into. It's a different target, right? Influenza is water birds mostly, and I guess pigs and chickens. But this has to be done. Don't you think this needs to be set up on a big scale for SARS-CoV-2?
Yeah, although
Absolutely.
we're in a good position because we can sort of narrow down where it's needed.
Yeah.
Sort of. But I mean, it could be a simple solution. If it turns out that we're completely wrong and it's from the dog shelter, say, that's a solvable problem. You just have to quarantine it until it's cleared, and then any risk is gone. But you can't do that until you know where it's coming from.
So
we need resources and money to keep, just like we said last time, to make this kind of work happen. We need more hands, and we need more resources, more money. We just can't do it on our own. It's not possible. So we need to almost integrate with existing surveillance strategies in New York. So like you said, Christine is doing citizen science type approaches, and it's many hands lighten the load, if you like. But with this, because it's in the wastewater and it's in the tunnels, it's in the sewers, it's not so easy to do that.
Yep.
So to track back is extraordinarily difficult, and it takes tons of time and resources from the DEP to help us with that work. And they have helped us. We've tried it, but it's to do it all the time. It's mind-boggling.
Yeah, and I also think that in also picking up an idea that is not mine, but we are not doing sequencing all over the world.
No, we're
but
not.
We do know that the world communicates. So we detected Omicron in New York, right, on the 21st of November.
Mm-hmm.
Actually, almost at the same time that South Africa was talking about it. But the thing is that we don't know any other places where these things could be coming from because we don't have sequence.
Sure.
So
we detected it in New York City 4 days before it was reported in South Africa.
Yeah.
And 10 days before the first case.
And your samples or people?
The sample was collected 4 days before.
Yeah, the sample.
It was the week before Thanksgiving when we first.
That was a clinical sample. Is that what you're talking about?
No, in wastewater.
Wastewater. Before any clinical samples.
Yes, as far as I know, that not every clinical sample is in GISAID, but we had a wastewater sample that turned out to contain Omicron from November 21st. And the first patient sample from New York that I'm aware of was collected the day after.
And we have to assume that the patient samples, they knew where to look for those samples.
Yeah, that was
because they were tracking the patients returning and so on and so forth. So this process, what we're doing effectively gives us a much greater coverage all the time. Unbiased. So to Monica's point, we do have to give a plug though. Vincent, we're very grateful to you because myself and Monica, we actually secured a donation from a foundation that has given us the funds to support a project in Uruguay, where we're actually helping the folks down there. Monica's actually traveled down with supplies to help them do sequencing in Uruguay. So what we're trying to do now going forward is to try and identify, if we can, ways that we can support folks who aren't in essence in a position to do what we're doing in the north, because ultimately it's a huge, it just purely from a scientific standpoint, we don't know what's happening anywhere else. So, and this targeted approach, the way we're doing it, is relatively inexpensive. It's cheap. It's not as expensive as the expensive kits. So that's something to consider. But I also want to say that we also got a donation from Kaizen of kits. And so we've been doing whole genome sequencing as well to try and expand beyond just the receptor binding domain. So a lot came out of this podcast. So we're eternally grateful for you having us on.
So having started to learn, so we're a 501c3 now, we're a nonprofit. And so I am learning about fundraising. So there are several sources of raising funds. There are individuals who have a lot of money and they would like to give some of it away because it gives them a tax deduction. There are foundations, there are charities set up to do that. And then there are companies who have charitable divisions, and you can approach all of them, and they are well known to, I hired a fundraiser just to give me names and to help approach those. So I think you have a very good cause that would interest many people. So
we haven't talked about the antibody.
Yes, let's talk. Let's talk about that.
I want to hear you guys describe what those data consist of and what you make of it.
So that was my lab. Jesse I rely mostly on Jesse Blumen's he went through these papers where he systematically made every possible change in the receptor binding domain and then looked to see whether they would affect any of the monoclonal antibodies. And we looked and like, oh, a lot of these have the same mutation. When the first time I saw his paper, I'm like, this looks like a hand in the glove with the stuff we were seeing in wastewater. So it looked like they were going to be resistant to the monoclonal therapy. So we actually didn't get the antibodies. We ordered the plasmid from a company for making the monoclonal antibodies, neutralizing the antibodies. And we made the pseudotypes with and without the spike mutations from the wastewater samples. And then we just, did your standard neutralization assay to see whether they still worked. And at least with two of them, none of the three classes were able to neutralize anymore, which is the same as what they found with Omicron 5 months later.
Yeah. And that's, so you reconstructed fully the lineage RBD in those, right?
Correct.
There's not single amino acids, the whole collection,
no.
right? Okay.
Yeah. And there were some we went further where we actually were able to amplify most of the S1 domain, which would be even more effective because some of the neutralizing antibodies do target the NTD.
Yeah.
And those were even more. Sorry, I didn't get into it. We also did patient serum. I have a colleague here at the University of Missouri that has a large cohort of samples he's collected from infected and vaccinated, you name it. So we worked with him to check how it did with patient sera. Everyone was able to still neutralize, but especially when we included all of S1, they were way down compared to, that wasn't in the paper. That was something we did later. They were significantly down, but there was always still some neutralization left.
So when I look at those data, it suggests to me, perhaps naively, that these have undergone selection in humans and would favor the human origin idea over the animal origin idea.
Well, animals make antibodies too.
Yeah,
and
they do.
And, an antibody is an antibody. The ones that can neutralize are going to neutralize. So it doesn't matter which animal it came from.
Yeah.
That's what I always wondered about this, though. If you have the same virus in an animal and in a human, and the evading antibodies, do you expect the same RBD mutations
I guess that's the question.
Animal as you would in human?
That's why antibodies are not like evolutionary classes. They're just ways that an antibody can bind.
That's why we need to get access to as many different sera samples as we can. So that's why by working with the USDA folks, we were able to basically say, if you're looking for sera, can we have it? So we were hoping if they're looking for rabies and other types of viruses, could we just tag along and see if they're doing that? But it turned out that they were doing the deer stuff alongside what we were doing. So, but I think that's what's going to happen. We're going to have to start surveilling wildlife and just doing it all the time for this now.
So this would be a good time to mention, John mentioned this earlier, but I'd like to make a plug for it, is that one of the downsides of Omicron is that a lot of our primers for doing whole genome sequencing don't work anymore. There, if you look on the sequence, if you look on outbreak.info or any of these things, they still don't have K417N as being a consensus site in Omicron. And that's not because the mutation isn't there. It's because more than 50% of the sequences in the database have a dropout in that position because the primers that they don't work anymore. But we decided to make lemonade out of the lemons. And what we've done now is we've designed a primer set that will only amplify virus that is not Omicron. So we basically have a way now of asking what is bubbling under the surface.
Okay.
So now that Omicron is the only human virus, it's going to be like last summer again, that the only thing we will detect is the lineages if they're present. So we've been reaching out to, I think I've emailed about 25 states so far and asked them if they would send us at least a single sample from each of their major sites. So we can figure out where else in the country these lineages might exist, because we know it's not just New York. We know that for a fact now, but we don't know how far it goes.
So you want to look at wastewater samples, is that right?
Yes. Groups doing wastewater. They want to send me a sample, we'll amplify. The only thing that will amplify will be non-Omicron lineages. So a lot of places what we're finding is they still have a little bit of Delta around. And as it turns out, the primers can still kind of amplify BA2. So a few places we found there's BA2 where they didn't know they had it because it was just bycatch from the assay. But if there's wastewater groups out there that would like to participate, shoot me an email. We're trying to get a good sort of coverage while this wave is still going strong.
Weren't there some wastewater studies out of Europe some time ago, right? And did they ever see any of these cryptic lineages? No.
No, I've written some of them and even had them go back and look. But most of them are doing whole genome sequencing.
Oh, that's the problem. Okay.
And most of them are looking for the variants of concern.
Yeah.
They are looking for, they have a reference, the wild type and then other variants. And whatever is different from that, it's thrown out. So it's a different approach that they will not see it.
Okay.
So back on the antibodies for a second, if there's this question about which organism is the host, whether it's animals or humans, could you actually use this information and say, well, do any of those potential animal reservoirs actually make one of these as their dominant antibody type?
Yeah,
and use that to sort of say, hey, it turns out that rats always make these antibodies.
Yes.
So they would be likely to lead to selective pressure to avoid this particular antibody.
Mm-hmm.
So
that was kind of the subject of your previous TWiV, right?
Yeah.
361, I think, with the mouse paper from a group in China published in Journal of Genomes and Geology or something.
It's kind of tough because it has mutations that are evading all three classes.
Yeah.
So it's pretty much got the bases covered.
So it could have been stepwise. So there's no reason to suggest that what we're saying, it could have gone from humans into animals and back to humans again. Like, we just don't know when those mutations were acquired. So we need a time capsule to go back in time and figure out where these first came from. So
it's strange though how they always seem to appear fully baked though, if you will.
I know.
Same way with Omicron, how it went and then it appears.
Yeah.
It was the same way with all of our, even when we see a new lineage, we've been sampling for a year and there's some we didn't really detect until this fall. And once we saw them, they were already fully baked the first time we saw them.
Yeah.
Well, I think that's a sampling issue. You're not above the limit of detection until it's in a lot of people, but it certainly is percolating out there and you just can't find it. It has to, right?
Yeah.
Well, if it's people, it wouldn't be just in one sewer shed.
No, I'm just talking about any variant like Omicron.
But he's just saying like the frequency of the sequence.
Right.
Yeah.
But if it's coming from a population, maybe it gets to a point where, it reaches an outbreak and whatever animal population reaches the threshold. That could be.
Well, that's why, as you've said, you need to be doing a lot of sampling. I mean, you're doing it in New York, but what's the chance that something's going to originate here? If it does, you'll see it, right?
Yeah.
You're going to see it.
But that's not true of every city. So
yes, no, exactly right. As you say, we need to do more in many cities, but I don't know. Do you have a sense whether that's going to happen or not? I mean, I just don't know.
Well, I'm just to give a shameless plug. So I'm new to San Antonio, so that's what I'm trying to do here. But like the folks in San Antonio, Vikram Kapoor over at UTSA, he was doing the the quantitative PCR for the whole year last year, but he didn't do any sequencing. So at least we can go back through the back catalog and see what was happening there. But I think right now, it's like anything. It's the resources and the hands and the manpower to do it. And why would you do that? Because for many labs, certainly the academic labs, like all the students in John's lab and everybody else, we've been doing that and, not doing the other projects, the master's projects and the PhD projects and everything else. So in order for this to work, we need to have a mechanism by which we can train the environmental labs to do the assays themselves, like we did with DEP. And that's going to take time to train folks to do that.
Sorry.
No, so the capacity for doing it is what's lacking right now.
I just wanted to comment on this whole idea of like it appearing. There was there's this one sewer shed that is different from all the others because the lineage is very clearly derived from alpha. Mm-hmm. All of the others seem to be much earlier, but there was one that was derived from alpha, and that was one where we only started to see it in October, November. But it's alpha, and one of the mutations it has is K417T. Very common to have a mutation of 417. But when we went back in April, one time we saw an alpha variant with K417T, and then never saw it again. So it very well could have been the early generation of this lineage, which we've now seen for several months.
So the other lineages all arose from an ancestral pre-VOC virus?
We think that the others are very early.
So why don't you see some derived from currently, like Omicron? It's hard to get the noise out of that, right? It's too noisy because they look like Omicron. Is that right?
Yeah, it could be. Yeah.
So we're
You
like,
remember the Omicron variant didn't come from Delta like everyone expected.
Yeah,
It come
right,
from
right.
the predecessor, right?
Yep. Interesting.
Kind of came out of nowhere from a variant that we thought had died away. I think it was Alpha, right?
So that's why we think it's possibly patients that are just bedridden, that aren't mobile, that are severely immunocompromised, and they're just chronically infected.
You haven't noticed, even among the group, we're not of consensus of where we
No, it's fine. It's fine.
And for the record, part of the reason for this was early on, and I think I don't know if we talked about this last time, but when we've recognized early on when they were sequencing the swabs from people's noses, that we got a couple of samples from humans that we did the sequencing on, and we identified that this it was not a uniform single virus in the people. So we saw these minority variants even among the individual sequences that we examined. And when we talked to folks, what they were doing was they were just effectively generating a consensus and uploading that. So that led us to this kind of consideration that even within an individual, there are additional variants that are contributing to the population. So consider that over a long period of time, patient is infected, they're sprouting multiple variants, and then you multiply that by the number of people that are living in New York, and it's a very, very big number. So I'm still convinced that it's going to be people.
But we did look at human SRIs that were available and did not find these.
Yeah, but I think, like I said, what we need to have is chronically sick people, sequencing them over the length of their infection and see the diversity of that. And a couple of groups were doing that, but we need more of it.
Have you ever done the thought experiment of trying to figure out how much virus a single individual would have to be shedding in order for you to pick up a signal in one of your samples?
Grams.
I think that's
grams. Yes, I've done
extremely difficult question to answer, because also it varies from wastewater treatment plant to wastewater treatment plant. I can only tell you that we did a couple of studies to look at how much inhibition there was in the qPCR in different wastewater treatment plants. And not only they differ between themselves, but also they differ in different time points. So it's a very good question, but it's very difficult to address it experimentally.
Even before you get to the wastewater treatment plant, you have a variation in individuals or how much they're shedding. Some people may not even shed from the feces. Other people shed quite a bit from the feces.
Yeah. But if you look at the copies, if you look at the copies per liter, multiply that times the sewer shed amount, and then you calculate in what percent appears to be your variant, and you do the math, it comes out to grams.
That's a lot of poop.
So do we have any estimate for how many immunosuppressed people live in the New York City area and how many of them are infected with SARS-CoV-2? No idea, right? I mean, these may not be people who are institutionalized. They may be at home and they don't even know they're immunosuppressed and shedding, right?
Well, and how immunosuppressed is immunosuppressed?
Yeah, maybe you don't.
About all of the people who are on Humira.
Yeah, anyone who's on a steroid, basically.
Yeah, sure.
So it could be anyone.
But frankly,
that's why we need to have higher resolution. That's what we're looking for. We need to get, instead of just having 14 samples, we need to have facilities and regions of the city and doing it all the time and not just once a day. There are so many different variables that you just can't account for them. But what we're doing right now is just a 24-hour composite once a week. And that's all we have.
From what I gather, though, the level of immunosuppression it would take to account for this is most likely either an unmanaged HIV patient or an organ transplant.
Yeah. Severely
there are a few others,
immunocompromised.
but that covers most of them. And actually, the health departments do have reasonable records in most places for those kind of things, but it's still tracking it down from a top down is it takes a lot of manpower, and it hasn't occurred yet.
And that's not exactly been forthcoming. Part of the problem is, we get the same response from a lot of people. It's like, that's really interesting, but then you don't really get any follow through to an assistant to track these things down, which is manpower, which is not necessarily financial resources, but getting the phone, contacting people on email. We've reached out to quite a few people by email, by phone, and outside of the academic community, we don't get a good deal of people responding to us.
But if you read the comments in the New York Times article, we're not being aggressive enough. We have to follow up with a letter.
Oh, yeah, that's going to make a difference.
Well, one of them said we have to go there and knock on doors.
No, that's not good.
Oh.
So you have
But
to
we
know
have-
you have to know the right people to contact. For example, have you approached the Gates? You probably sent them an email, but that's not good enough. You have to email a specific person at the Gates who's going to respond, right?
Well, we're going to get your guy to help us with that.
If it turns out it's people, I'm happier than it being in animals, right?
Yeah.
Yeah.
Because we can handle that. I would say all these people who are shedding, you could give them a course of antiviral and stop it, just like they did. Actually, the young lady in South Africa, they treated her AIDS, and that got rid of the SARS-CoV-2. So it can be fixed.
I thought they also gave her a monoclonal therapy for the SARS.
Yeah, maybe. I forgot. But, it would be interesting if we start such therapy of the cases in New York, and then you see the signal go away, right?
Yeah.
In the sewage.
That's very interesting.
Against your theory, I think one of the things I was also discussing in the program is that there are many mutations in the RBD of a cryptic lineage, something kind of similar to what happened to Omicron. And when you look at some of the data, there's not a lot, but some of the data coming from immunosuppressed individuals, they do have mutations, but they don't have that many. That's the part that it's a little bit hard for me to understand. What is happening that suddenly you have this huge number of mutations in the RBD? I don't understand it.
Yeah, we should mention some of our lineages have 50% more mutations than Omicron. It's
So you never see that much in an immunosuppressed person. You never see that much.
But think of it this way: if we were able to get fecal samples from immunocompromised patients across New York City, how fantastic that would be. I mean, and we're begging for them.
Well, you have
and
to,
when I say we, I mean me and the New York crew. But that's what I'm saying. If we could get access to those kinds of specimens, and if you think about it, like it's poop, so it's being thrown away. Like it's bedpans. It's not like we'd be asking for any. It's not an invasive specimen. It's just a poop sample.
Yeah.
So,
well, you have to hook up with the DOH, right? They're the ones who would do it. Or you could find people we have a lot of big hospitals here in the city. You could find people who are interested. You could hit the chiefs of infectious diseases and, you could send them this episode and say, hey, help us. I think that would be really useful.
Sounds easy, but it hasn't been.
No, I didn't say it's easy, but there are ways to target individuals who might be interested. Before Scott Hammer died, he would have been the guy at Columbia. Scott, look, can you help these people? And there are people at other institutions who have similar positions that you find someone who knows them and get them to ask or connect you. I think those are the people to help you. I mean, the Department of Health is hard because, New York City's a mess. I work here, so I can say that, but individual hospitals might be a good source of this material. And so I would try and contact people. And a guy like David Ho at Columbia can be very useful because he's connected to everybody. And it's just a matter of sending him an email and saying, we found this, and he may be very interested or not. So that's the way to do it. I mean, I'm not trying to do your project for you, but that's my suggestions. It's doable.
Can we CC you on the email?
Yeah, I mean, I can email David and put you guys on and say, talk to these people about this idea. Give them some poop. Yeah.
Well, I want to tell you a funny story. So when we were turning on the lights down here at Christmas time, they do this thing where they turn on the lights for holidays or what have you. And they had a petting zoo. Petting zoo.
Yeah.
Now, I've never seen a mobile petting zoo. And they had like porcupines and they had like, I don't know what they're called, wallabies, all kinds of tropical animals. And Davida, of course, was like, can I get their poop? Because it's a petting zoo, so they've been extensively handled, and not only that, they're in amongst the cages with one another the whole time. So I got that woman's card, and it's just a matter of time. As soon as they let me do it here, I'm getting me some wallaby poo.
By the way, I don't want to give you more work, but you know, you can look for some other viruses in your specimens easily, and
of course,
yeah.
You should look for polio virus because I'm sure it's all over the place in New York. Have you looked for polio?
We haven't looked for polio, but Monica has looked for flu.
I'm sure you find influenza virus. But polio virus is interesting because, as you know, there is no paralytic polio in the U.S. It's been eradicated from the whole Western Hemisphere for most countries except, but I'm sure the virus is in the sewer here. It's probably vaccine, but it could also be wild virus because the vaccine, folks, does not prevent infection. Like other vaccines do not prevent infection. And so people shed virus. They come in from countries where the virus is circulating. So, you know, I once asked CDC, why don't you look for polio virus? And the guy said in the sewage in the U.S. And he said, because we will find it and then we don't know what to tell people when we do. I think that would be fascinating. You'll find it.
We did shotgun sequencing on our Missouri samples a few times, and we didn't see polio, but we saw a lot of other interesting things. Some not expected.
I think it's just because New York is such a melting pot, Mark. You're going to see everything here.
Well, we can try it.
It comes in.
I don't think wastewater testing is going away, so now is the time to figure out what else we can screen for.
Yeah.
I'd be shocked if you didn't find it. It would be very interesting if you did not. But anyway, that's my 2 cents. All right. Anything else, folks? We kept you over, but I think you don't mind.
We don't mind at all.
Anything we left out? Anything
for you guys? So occasionally we see what we call reference sequence in our sewer shed. We're not quite sure where this is coming from. By reference, I mean basically wild type virus. What do you think are the chances that we could be detecting people doing PCRs and the biohazardous waste somehow getting into the sewers? If you're doing not the rapid test, the PCR nasal swabs and companies basically testing in New York City, dumping their waste somehow.
Let's explain a little bit more. What happens is that sometimes when Marc does the sequence of this small RBD, he gets some pieces that have no mutations, that it looks exactly like the Wuhan, right? So our first and still perhaps a possibility thought was that we had a contamination.
Yeah, that's what I would say.
And so we immediately looked at, what are we using as a standard? We are using as a standard EURM that doesn't have the RBD. We haven't used TWiV for the last 6 months. We have done experiments where we have multiple method blanks where we do not detect it. Okay. And the only other piece that we can share is that we only detect this wild type form is when the levels of the copy numbers of the virus are low. So we never see it when, we have high numbers. And to be fully transparent, when Marc prepped some of the same samples that we prep here, he didn't see it, right? But just let you just be aware that these are samples with low copy numbers. So whatever you amplify, it's not really, it can be stochastic. So you get it or you don't get it. So we've been struggling with this.
So what I'm wondering is basically when you run a PCR plate, you use a positive control. Is that positive control going into the wastewater?
Yeah.
And if so, could we detect it?
It's possible. I mean, when people grow up plasmids and E. coli, they lyse it and they throw it away, right? They probably put it in the orange bags. That's what they're supposed to do. But maybe some of that gets into the sewage, right? I mean, I don't think there's a lab on Staten Island that would be doing that. Maybe you could see if you never get it on Staten Island, that would be consistent with it, right? But certainly Manhattan has plenty of biolabs. The Bronx, I don't know. Would there be some biolab?
Yeah, the Bronx has Einstein and the hospital.
Einstein, of course, yes. And Brooklyn does, Queens. So I bet, Because we had some years ago, at the beginning of the pandemic, we had some false positives in people at Columbia, and every one of them was working in a lab that had the plasmids. And I'm sure
wow.
they got
I
into...
hadn't thought about the plasmids. I was originally imagining qPCR, and I was having a hard time
I think the plasmids...
connecting it into the wastewater, but the plasmid makes sense.
Plasmids in the sequencing lab.
Do you guys remember the story of a couple of guys or a little bit more that were either at Harvard or MIT that were working with plasmids, with part of SARS-CoV-2, and they were being tested, and they were given a positive test.
Sure, yeah.
And they were not infected, even their backpack had positive signals.
Yeah, the plasmids get everywhere.
I remember that.
You pipette, it aerosolizes, right? It goes in the air, and you breathe it in your nose. You go for a nasal swab.
That's what I always. No, but that's what I always loved about when we first started sequencing, Monica, and all the companies will tell you, you're going to have the sequencer in this room, and you're going to have your PCR machine in a different room, and you're going to have everything separated. And then we're like, but we only have one lab, and we don't have the luxury of separating everything. So plasmids and the sequencer.
The thing is that in this lab, we don't have any live virus, and we don't have TWiV.
Plasmids.
That
and
is the one, and we don't have plasmids. So
I'm sure it's plasmids.
That's why it's so hard to figure out.
I mean, the gel boxes that you run gels in, they're totally contaminated with plasmids of all sorts that you've ever run in that gel. And you rinse it out in the sink, and they all go down the drain. So I'm sure that may be part of it.
I'm thinking about Eric Delwart the whole time during his conversation.
Yeah.
DNA is really a problem. It's everywhere.
It's a real problem, yeah. I'll bet that's it. That's very interesting. And it just shows you how bloody sensitive it is, right? All right, folks, this has been great. Let me say goodbye from Queensborough Community College, Monica Trujillo. Thank you so much for joining us. I guess we'll see you in another year, right? You'll have some more results.
Yeah. Once we find it.
Yeah, or as soon as you find it, call us.
What if you actually spiked a plasmid somewhere and then tried to see if you could detect it downstream?
That's all I was thinking of. I read an article about barcode synthetic DNA and how they use it to trace things through the
what if you went to an apartment in one of these sewage catch zones?
I'll just go to the dorm and flush it in the dorm. And look if it comes out the dorm water.
They did that with polio in Japan. They used to immunize 3 times a year, oral polio vaccine, and then they would look in the sewage, and X days later they would see the virus in the sewage.
Wow.
And you can find it, yes. They've done that in Europe as well. You can easily trace that. So, yeah.
I really like the spiking experiment.
That's a lot of people, though. When you give OPV, you're giving a lot of people. So you spike.
Yeah, but it can tell you, the limit of detection.
You have to spike a lot.
That's brilliant.
Yeah.
Yeah, some plasma. But anyway.
When we find it, what are your predictions? What do you think it's going to be if we find it?
What do you mean?
What are the cryptic variants? Where are they coming from? They're coming from rats, cat, dogs, something else.
Okay.
Someone
No, I like the humans thing. Yeah, human and immunocompromised humans. Yeah, and it's so localized. It seems just the more logical source. The thing that gets me more than anything else is that it's got to be a lot of virus coming out of a few people.
Yeah, that's true.
So,
John Dennehy from Queens College. Thanks, John. Good to see you again.
Good to see you too.
From Texas A&M, Davida Smith. Thank you.
Thanks very much for having us.
Much better digs this time.
Yeah, it's bigger. Everything's bigger in Texas.
That's what I've heard. So you're working to try and get San Antonio doing wastewater.
Yeah,
sampling,
and it's a completely different system here because it's 2 companies. Actually, it's private down here. We've got this crowd called SAWS and this crowd called Sarah. So I'm dealing with companies now as opposed to DP. But it's so far so good. They're very interested.
Oh, good luck.
And I've even persuaded some of them to come into the lab and do a gram stain with me. So baby steps.
Nice.
And from the University of Missouri, Marc Johnson. Thanks for coming.
For having me.
Yep.
Hey, and so no one else voted. Where do you think they're coming from? Anyone want to vote?
Go ahead, Brianne.
I'm struggling. I think that the localization does definitely argue for a human source, but that much?
I was just going to say, remember Typhoid Mary. You all have to remember Typhoid
Mhm.
Mary.
Yeah.
That's all you have to think about.
Yeah, but she was shedding a lot of bacteria, right?
But she was a super shedder, so it just takes a couple of super shedders. And she didn't know she was sick, remember? She was asymptomatic.
Would you pick that up in sewage, though? Because they were picking it up by other people getting sick, right? So
We'd have to go back. We'd have to go back in time.
That's a
Need
big
to do the
difference.
spiking experiment. Brain
Exactly.
spiking experiment. How much does it take?
I think it's a
I mean,
brilliant idea.
how much are you going to try up to? Are you going to put a gallon of plasmid in?
Until you
know?
can see it.
Well, like Marc said, we're going to be dealing with grams, maxi preps.
Yeah. I don't know. I think I'm leaning towards people also.
I want to point this out. One thing that's sort of still I can't wrap my head around is like with Omicron.
Yeah.
How could there be two? So I can, BA1 and BA2 are related, but if it was in a immunocompromised person and it spilled over, why did both versions spill over and then separate? It's kind of hard to wrap your head around how that would have happened unless they spread it to different people at different points in their infection. It just
the problem is, Marc, that we really don't know what the selective forces are. We think we do, right? We think, oh yeah, it's antibodies, but you just talked about a tiny piece of a huge genome where there are other changes that you're not even looking at that might make a big difference too, right?
But regardless of what the changes are, it spilled over twice from wherever.
Yeah, I think that's probably what it was. It spilled over twice and then went independent selection. I'm okay with that. I mean, you just can't figure it out. That's why you're puzzled, but you're not going to figure it out probably. But
I think
it's just under that circumstance, for me, it makes a little more sense for an animal reservoir. But we'll all find out eventually, I hope.
maybe.
I would say that before this episode, I was leaning towards animal reservoir, and honestly, right now, I'm leaning towards my head hurts.
All right.
I
All right.
change my mind every hour.
Okay, everyone. Thanks for coming. Appreciate it. See you next
Thank you
time.
very much.
Great talking to you.
Bye-bye.
Thanks. This is great.
You all.
Oh, that's cool.
So what are you going to do? You're going to take a plasmid for what? With some marker that doesn't exist anywhere that you
It
can...
doesn't even matter what it is, but it's got to be absolutely unique. I mean,
It's got to be
Yeah.
unique.
It could be
Yeah.
SARS-CoV-2, but
No, you don't need to
somehow
do that.
barcoded.
No,
Yeah.
don't do SARS-CoV-2.
No,
that
no,
would be
no, no,
bad.
no.
No, it's too confusing. Yeah.
No.
So, but you got to find something that's unique. So,
PBR 322. How much?
no, it's not unique. People still use it, right?
Yeah.
Well, whatever it
Yeah.
is, you have to test it in the sewage before you release it to see if you get a signal. And if there's no signal, then you could
Right.
do it. And
Right.
where are you going to do this? From what part of the city, Brianne?
I don't know. That's why I don't know enough about what the city's doing. I actually like the idea of doing it in a dorm.
I think you want to do it from probably several different input points
Yeah.
from one of the treatment plants where you're seeing the signal.
That's right. One of the,
Okay.
and you know where it's
And try
going to go.
several
Yeah.
different points.
Yeah.
And you're going to have to do a titration.
Yeah.
As a matter of fact, might have to titrate with different sequences.
Yeah.
So that you can distinguish one from another.
But of course, you have to be completely transparent and you're going to have to get permission to do this
Yeah.
Sure.
from the university or hospital, wherever it is that you do it from the city. You can't fool around because this can't come out afterwards that you did
No,
this
definitely not.
on the sly. And they say, oh, we're going to release a gram of PBR 322. Good luck with
So
that.
this episode, I think, is a great example of science in action. You get to see that was a really nice story of how these people got together, their individual enthusiasm for the project and their different approaches to the project. And then, I mean, we don't have the answers, right? So they're arguing with each other and thinking on their feet. And it's just great. It's perfect.
Yeah. Then also, so they're willing to share this. You know, not everyone wants to do this
Mhm.
in public. They want to just do it in their offices and via Zoom, but they want to share it. But also, what I love is that the city was going with somebody else and they couldn't get it to work. And these guys got it to
Right.
work.
And these
I
guys
love
did
it.
it better.
And this is, you know, a local university, which I think is great. Plus, it's evolving, right? It keeps changing. The story gets better and better. So, yeah, it's a lovely story.
And they learned cool things.
For sure. I just am sad that they can't get support for it, right? They need to get support. And, you know, I see big labs getting bigger and bigger, and it's not right. They should get money to do this, right? That's it's kind of the beauty of science, and the sadness of it is that it's really hard to get money to do stuff. It's too bad. But yeah, it's a great story. Glad they're here. Let's do some picks. Brianne, what do you have for us?
I have an article that I read that gave me a lot to think about. It is called "When Scientific Conferences Went Online, Diversity and Inclusion Soared." So this is a report in chemistryworlds.com that is in fact talking about a study done by others that talked about when different conferences went online, the changes in attendance of different groups. And so, for example, at virtual meetings, attendance by women went up 253%. Attendance by scientists who are LGBTQ went up 700%. And attendance by students and postdocs went up 344%. And you can also imagine similar numbers, though they're not quoted here, in terms of individuals from maybe countries where they can't always travel to some of these conferences, or disabled scientists or things like that. And I think it's a really interesting thing to think about. I have sort of mixed feelings. I think that the diversity and inclusion is awesome. And I can personally talk about things I gained from some conferences being online that I never would have been able to go to. And yet I also really miss in-person conferences. I also think the environmental effects of everyone not having to fly are great, but I really miss some in-person conferences. And so trying to figure out ways that we can keep some of these benefits, even as we also try to get some of those benefits from in-person conferences, is kind of an important thing. And I'm glad that we are talking about all of the benefits of virtual conferences.
So is this because virtual conferences are more accessible to these various people, or some
I
other
think
reason?
Part of it is that you don't have to pay to travel. In many cases, the fee for the conference is less. And so that's part of it. One of the things that was actually really helpful for me was that there was a conference I found really interesting that happened during the semester, when I never could have traveled because of my teaching, but I could watch the talks sort of around classes or at night. And that conference was one of the best I'd gone to. So I think that there are probably lots of reasons. You can also imagine what some of them might have to do with things like childcare or other family obligations or things like that. But there are some really big benefits here. On the teaching side, I know teaching hybrid is sometimes really hard. So I don't know about hybrid conferences. They're probably pretty hard in some cases too. But we should remember these benefits.
I think probably cost is not one, because that would give an overall boost to everybody, not just those groups that you
it's
mentioned, right?
Apparently.
But I think that some of the individuals in different countries might be getting the boost, and that could be a reason why there's a boost for postdocs and grad students.
Yeah, maybe, but I think LGBTQ boost is not monetary.
Right.
Women is not monetary, which brings the question: is there an overall boost for everybody? Right? What about the other people that weren't included? Did they get boosted equally, and then everybody gets boosted? Right. So I'm not sure it's selective. Right.
Right.
That's the question. That's what I would ask.
I am not sure of the exact answer to that, but I think it's pretty cool that there are these increases.
Yeah, I think it's great. And I think being able to attend when you're teaching and you couldn't travel, that's wonderful. And we've learned that during this pandemic. I just don't know I think it's probably applicable for everyone. I'm not sure why an LGBTQ, you think they might feel uncomfortable going to a meeting in person, right? And that's part of it?
Perhaps.
And same with women. I could see that, right? They're intimidated.
Yeah. I wonder how many women weren't necessarily traveling because they needed to be home for family obligations.
Yeah, I'm sure that's it. Yeah,
you're absolutely right. I don't have a great explanation for the LGBTQ.
I think, though, that I agree. It's a great outcome. And I'm sure there's going to be a combination, right? Conferences should consider being hybrids, right?
Yeah.
I mean, my class now is in person, but the students are always saying, is there a Zoom option? Could I stay home? And maybe that's not a bad idea. I get way more questions on Zoom than I do in person. I said the other day, maybe I should put a Zoom chat room up so you guys can ask questions who are too shy to ask in person, right? So this online stuff is good for many reasons. But, you can't drink beer over Zoom.
Not in the same kind of way, no.
Or whatever. You can't go golfing, Rich, over Zoom.
Right.
Well, you don't have the same kind of sitting next to somebody and randomly start chatting and make the connections.
Yeah.
Or sort of get some of those interpersonal interactions that I found helpful.
They're totally different. You can't be arguing that they're the same. I agree with that.
No.
When I first met you, Brianne, we were sitting in the audience at an ASV meeting, and this has happened before, and I think you heard me.
I think so. Yes.
Behind you.
I think so.
And you turned around and introduced yourself. Okay. As you had just started with TWiV. Okay.
Yeah.
That kind of random stuff is not going to happen on Zoom.
Right. Exactly.
No.
But we should at least, before we sort of throw out the Zoom, we should make sure we're keeping track of the benefits.
We're not going to throw it out. It's not going anywhere for sure. It's been proven.
Oh no, this is a big. This is a change.
Look, the trains into New York are empty because many people are still working at home, and companies have learned they don't need to pay for big office buildings to get productivity. People can work at home, so they're not all going back to work. So it's here to stay, and we're going to figure out how to work with it. I think it's fine. Cool, Rich. What do you have for us?
Okay, I have a little amusement for you. This is a YouTube video called "Traffic Flow Measured on 30 Different Four-Way Junctions." So this is modeling of traffic flow, and these are animations where you're looking at different kinds of junctions from an aerial view, and they're all four-way junctions, starting with just a four-way junction with no traffic lights or anything, and asking how fast the traffic moves through this, going through these extraordinarily complex interchanges like a stack interchange. It starts off with a four-way road having a something like less than 100, I think it's cars per minute or something like that. I forget what the units are. And then the stack interchange is like 1,000. This interested me, among other things, because every time my wife and I are sitting there at a light, we start talking about how a roundabout would be so much more efficient.
Yes,
okay,
indeed.
But it turns out, when you look at this, that's not necessarily true in all cases. The roundabout is not necessarily faster than a four-way interchange with dedicated left turn lanes or stream right turn lanes or something like that. At any rate, this is crazy. It was put up I think was put up in 2017. It has 24 million views and 15,000 comments, and the comments are about the best part. Just for one example, a comment: "Now you know you've reached the time to go to bed side of YouTube."
I like this one. The guy goes, "I like how drivers would rather die than go through the grass." Of course, this is a simulation. The funny thing is, without traffic lights, it's a mess. But they have the cars just going through each other. It's like, because it's a simulation. That's a piss. Oh, they have a roundabout too.
Yeah. The roundabout,
and it's
a
not
straight old roundabout is surprisingly inefficient.
Interesting. Wow, that's pretty cool.
Yeah, I enjoy this.
That's fun.
Nice. All right, so I have a book that I have read a long time ago, and I'm slowly moving books from my Columbia office here. So this is an autobiography of Salvador Luria.
Oh, great.
A slot machine, a broken test tube. It was very thin. So, hey, guys, you can read it quickly. And there's Salva on the back. And this Luria, very important character in virology. And this is his story. He was born in Italy. He's Jewish, so he left, as the World War II ramped up. But he came to the U.S. and started collaborating with Max Delbruck on bacteriophages. And perhaps his most famous for the fluctuation analysis, which showed, at least in bacteria, mutations arise in the absence of selective pressure. They just occur, and this is what we understand today. And they did a very simple experiment where they plated phage on lawns of bacteria, and if it's influenced by the bacteria, should be a standard number of resistance on each one, and they're not. They're random, and so very famous experiment. I think that's the slot machine there, part of the title. And they got the Nobel Prize in part for that experiment in 1969, Delbruck and Luria. So it's a very interesting autobiography starting from his origins and how he got trained and went to the U.S. and helped start the phage group, which led to the modern molecular biology revolution. So he ended up at MIT. He was the director of the Cancer Center. So they made a cancer center at MIT, and that he hired David Baltimore, Phil Sharp, Bob Weinberg, among others, to populate that cancer center.
Wow.
And I did my postdoc there in that building, and his office was on the first floor, and there was an administrator there sitting outside his office, and I had to go talk to her for about a few administrative things when I first went. And one time I'm talking to her, and he walks out, and she said, "Meet Vincent Racaniello," and he said something to me in Italian. So what he said was "Di dove viene," which means "Where do you come from?" And I misunderstood. I was nervous because I knew who he was, and he's a freaking giant right in the field. I was just nervous. So he said, "I'm fine, thank you. How are you?"
Oh, no.
And he looked at me, and I'm thinking he must think I'm adult. I'm sorry. I finally got it, but he just had that way of staring at you, right? Anyway, that was the only time I met him. I never saw him after that, and he passed away some time ago. Anyway, it's a good book. It's worth checking out.
That collaboration, he and Delbruck, is, one of a kind.
Absolutely.
Yeah.
Absolutely, yeah. I mean, it's funny, and so he met Delbruck, and he was at Columbia very briefly when he came to the U.S., and he said, we spent 2 days doing phage plaque assays. And someone I know said, "Oh, that's cool. They did binge plaque assays for 2 days," which is probably what it was. All right, TWiV 864, show notes, microbe.tv/TWiV, questions, comments, picks, TWiV@microbe.tv. If you like what we do, help us out financially. Go to microbe.tv/contribute. Your contributions are tax deductible, at least here in the U.S. Rich Condit's an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.